Hi, friends. Sorry about the long wait up. There was one. The computer again took forever to tell me it was recording, and so I don't want to start before it actually is. So this is part 2 of the cell notes. We're going to go through some of the organelles that really make eukaryotes special and make them unique in different ways. We're going to talk about how problems in some of these organelles can cause big issues inside our bodies, and we're going to talk about how they all kind of work together to make something awesome. So cell organelles, big one, the most famous cell organelle probably, I see it all the times on memes and stuff on the internet, and that's mitochondria. And so the mitochondria is the cell's energy center. You always hear it referred to as the powerhouse of the cell. The mitochondria's main job is to turn food into chemical energy called ATP. And because we're smarter than this now, we're smarter than what this is on the slide, it doesn't turn food into chemical energy called ATP. It turns carbohydrates into chemical energy called ATP. It turns glucose into ATP. And ATP is this chemical energy that basically is the battery of the cell. It's what's charging the cell. It's what actually gives energy to the cell. Carbohydrates we use to make ATP to do these things. And that's the job of the mitochondria. That's what it does. And we're going to learn a lot more about the mitochondria as we go along about cells, but they're quite fascinating, to be honest with you. They have so many unique principles that make them super special compared to other organelles. One of the biggest ones is they actually used to be their own living thing. Our cells back in the day, long before us, hundreds of millions of years ago, billions of years ago, in fact, it's called the endosymbiosis theory, that mitochondria used to be their own living thing, and our cells swallowed them up and just kept them alive because they did the job better than us. They made more ATP, and we used them basically as partners, and we still use them today. We actually have no capability or no way of making more mitochondria if they die in our cell. Mitochondria can only make other mitochondria. If you use cell theory, that kind of makes them a cell. Mitochondria can only come from other mitochondria. And they also have their own DNA. They have their own ribosomes. They're basically prokaryotic cells that live inside our eukaryotic cell. They're quite fascinating. So we'll talk a lot more about them in more depth. But for us right now, they are our powerhouse of our cells. They are our cell energy builders. Golgi apparatus. Golgi apparatus kind of looks like a stack of really gooey pancakes. We used to call them Golgi bodies, and so if you hear me say Golgi body, I apologize 100 times over. It's just instinct. Okay. So a Golgi apparatus, its job is to process, package, and export proteins. It's kind of like to a post office. That's what I always compare it to, or like a mailroom of a factory, or someone that takes everything, packs it up into like a box, puts a stamp on it, gets it going, and sends it out. And what it's sending out is it's sending out these little vesicles. We'll talk about vesicles in a second. Vesicles basically contain the things, the proteins, and it sends them to where they need to go, either in the cell or the body. So it's a mailroom, basically. It's going to take in what we make as proteins and send it out of the cell. Lysosomes. Lysosomes are probably the most fascinating organelle to me, even though they're probably the simplest. They contain digestive enzymes. They break things down. We call them suicide sac, but I don't really like that name. I like something called apoptosis sac. Apoptosis means programmed cell death. But why we call it a suicide sac is because these guys contain all of the enzymes to break down a cell and to break down any macromolecule that a cell can come in contact with. And so there are a lot of different situations where either accidentally or on purpose or due to an infection or a certain kind of poison, lysosomes pop and their enzymes get out and they start eating the cell from within. So apoptosis, APOPTOSIS, is programmed cell death. When your cell knows that it can no longer do its job and it has a replacement, it purposely goes through a process of popping its own lysosomes to break down its own self. So it breaks down its stuff into resources for the cell that's going to take its spot. But this lysosome contains all these digestive enzymes. Enzymes are proteins that help make things happen quicker, and they break things down pretty quick. Now, there are some genetic conditions where people don't break down or have defective lysosomes. One of the more famous ones is Tay-Sachs. Tay-Sachs, their lysosomes basically do not have the ability to break down waste, and their cells basically slowly fill with waste and don't have the ability to break them down, and their cells will eventually not work, unfortunately. And people that have Tay-Sachs usually don't make it past age 4. There's other situations with lysosomes that, like I said, a lot of venoms and poisons are designed to pop lysosomes in cells because it's a clever way of basically you just have to pop this little water balloon inside the cell, and all of a sudden the whole cell is going to go away. So it's a clever way to evolve poison or venom, but for the most part, lysosomes super important. They're basically the janitors of the cell. They're cleaning up waste. They're reorganizing materials and making sure we can recycle things that we can recycle. Endoplasmic reticulum. There's 2 different forms of endoplasmic reticulum. Endoplasmic reticulum in itself is a large word, and so we're going to start calling it just ER. E for endoplasmic, R for reticulum. And what it is, it's a cell transport system. And so it's basically a highway to connect different organelles and make things move faster. It also can help a little bit with speeding up the process of putting the right parts next to each other, if that makes sense. Almost like a factory line or assembly line. And so rough ER, they contain many ribosomes. That's why it's called rough. There's a lot of bumps, and as we learned before, ribosomes make proteins. And so the rough ER is very famous for making proteins and then setting them down towards the Golgi apparatus to get packaged up. The smooth ER has no ribosomes whatsoever, and that's because they're more designed to make lipids, to make non-polar items, especially for the cell wall and cell membrane of the cell. So if it's an animal cell, it's cell membrane. If it's a plant, cell wall. So the smooth ER making lipids, rough ER is making proteins. We know it's rough because of the ribosomes on it. Smooth has no ribosomes, so it looks smooth. You kind of see the picture here. Both are super important. Most of the time, they both surround the nucleus. Like I said, it's an assembly line. It's designed to make things go quicker. And so they get the commands from the nucleus. They start making the proteins, and then they send the protein out through the highway. Cytoskeleton. Cytoskeleton helps maintain the cell shape and supports the cell, and also helps with the movement in the cell. When we watched the video, you guys saw a motor protein moving things that were walking on the actual cytoskeleton itself. There are 3 major things that we're going to learn about. 2 of them are cytoskeleton. One of them is the thing that helps make them. So microtubule is a hollowed tube. Kind of obviously think about the fact that it's called microtubules. Tubule means tube, micro means small. Microfilament is more like a string, and micro string, so small string. Both are used back and forth a little bit as different components within the cytoskeleton. Microtubules usually are used more for structure. Microfilaments are more used for movement to get from place to place. Centriole is a very fascinating organelle. It's only found in animal cells. This is always one of my favorite questions to ask because it's one of the only organelle that's only found in animal cells. And the centriole itself is used during cell division. We've watched it being used during cell division, but weirdly enough, when we take it out of the animal cell, cell division can still happen. So it's not as good. So centriole is definitely organizing and making it better, but it's not super, super, super important compared to some of these other organelles. If there's one organelle where you could have some problems with and still get away with it, it's centriole. The other interesting thing about centrioles is they help make microtubules, but then at some point, your cell stops making microtubules and just starts recycling them. So there's like a certain amount that it'll make, and then from that point on, it's just going to recycle its used up parts to make more microtubules. All right, just a few more here. One of the big parts of cytoskeleton is creating structures that allow cells to move. I just want to make sure I'm not going too far here. Making sure cells can move, and there's two famous ways cytoskeletons are produced to do this. One is a flagella. It's a whip-like tail here, and it allows for movement to go forward. And if you move it, it goes this way. Cilia are hair-like structures everywhere around the cell, and they have a very similar kind of thing. Their idea is they're going to spin or row in a certain direction to move the organism. Obviously, a flagella is probably more powerful, but it usually only works in certain directions, and it's hard for it to turn. Cilia, it's not as powerful, but you have a lot more range of motion with the cilia. That makes sense. So flagella, cilia. We have in some of our cells flagellas in humans. Cilia is a more common thing. We use cilia also to help us move particles in airways and stuff like that. And we can push things around. Also, cilia is used to keep our balance. You have cells with cilia inside your ears, and there's a fluid, and how the fluid hits the cilia kind of gives you a sense of how you're balanced. So they can have other roles too, but they're cytoskeleton that's exiting the cell to help movement or other senses. A vacuole. So a vacuole, or we sometimes call it a vesicle, same kind of idea. Vesicle, I used the word vesicle earlier with Golgi apparatus. A vacuole is a storage area for water and other substances. Animals have a ton of vacuoles and vesicles. We call it vesicles usually for animal cells. They have a ton of them. They're almost everywhere. They're being used a lot. Plants have generally just one giant vacuole, one huge vacuole. It can take up even 95% of the cell space, and it's full of everything from waste to things it's going to use. It's compartmented. It's like a giant storage area. So it makes sense. Plants like to store more because they don't know when the next time they're going to get water or food or anything like that. And so they store. While animals, we have smaller storage because we're moving around trying to find stuff. Last but not least here, I want to talk about is protein production. So the main job of a cell is to make proteins. Main job of the cell is to make proteins, because proteins are the building blocks of life. They make up what a living thing is. And so almost everything inside the cell is designed towards this process. We make energy to make proteins. We have DNA because it's the code to make proteins. We break down things in lysosomes so we can be more effective at making proteins. And so cell is a protein factory. It's a good way of looking at it. So the first thing is the DNA has instructions to build the protein. Those instructions are given to the ribosomes, and they make the protein. The ribosomes build the protein. They send it through the rough ER, the highway, and the protein then goes to the Golgi apparatus where it's packaged and sent out for export. Our cells are designed to make protein. That's what they're designed to do. And so everything in the organelles usually go towards this process. So this is part 2. Tomorrow we'll finish up in part 3. I hope it's making sense. We'll keep working on our drawing and other assignments in class to make sure that all these organelles are clicking in our brains. Thank you guys for listening in. I'll talk to you soon.